From philosophy into science, as knowledge advances.
All right. Hello, everyone. Thanks for showing up today to Proteins and Protein Folding 8. So today we're going to continue looking at transport proteins. And last time we looked at active transport. And of course, we're going to repeat this a little bit at the beginning of this workshop and also talk about today's main topic, which is passive transport. Right? And in particular, we're going to be looking at how passive transport is necessary for brain function. Okay, so let's hop right in. So last time we talked about a little bit transport proteins in general, and we found that there are two different general types of transport proteins. Some of them are active, meaning that they require energy to function and to transport proteins or transport things over the plasma membrane. And there are also passive transport proteins, which just work, for instance, using the concentration gradient. So if you remember, if you were here last time, we looked at the sodium potassium ATPase. And again, like the name tells us, well, of course, it is a transporter for sodium and potassium. And it is an active transporter because it uses ATP, and it splits off a phosphate of ATP and uses this energy to transport these ions across the plasma membrane. Right? We basically looked at the model last time, and if you remember, perhaps we had these transmembrane helices, and we had this ATP binding pocket inside of the cell, and if ATP binds, then basically the affinity for sodium is high, and sodium binds inside, then the pocket closes, opens on the other side, sodium is released, then potassium goes in, then the phosphate from the ATP is basically split off again, and which leads to recovery and potassium being able to go inside of the cell. A little bit a complicated process, but don't worry. If you understand the main mechanism, then you're all good, right? So I showed you this also last time, so this is mainly what I want you to remember from all the details that we talked about, which is just a short video about what it actually does. So let's watch it again one more time. Right? So this is the active transporter in its empty state, and it takes 3 sodium ions. It binds ATP to the hydrolysis, it changes its shape, it opens to the outside. Sodium is then released. And then afterwards the potassium comes, binds into the same sites. And when the phosphate comes off, basically the original state of the transport protein is restored. And thereby the potassium can go inside the cell. Right? It's also important to notice that basically what's happening, you're transporting 3 sodium outside and only 2 potassium in. Right? So if you think of the molecules as plus, you would see if you have a membrane and if you compare the inside and the outside, the longer this transport protein will work, the more you will have a difference in basically positive charge on either side, right? Because if you basically continuously put 3 on the left and 2 on the right, what you will end up with is with more plus on the left side, logically. And this is why we looked at it last time, because it's a fundamental state which is achieved in the axon of a neuron. Right? Actually, let's come back to that in a second. This one I want to show you. Right? If you look at the nerve cell here at the top, right, at the neuron, and you know neurons have like this one longer extension of like an arm, which reaches out to other neurons, which is called an axon. And this axon is like a tube, and you have a plasma membrane, of course, which makes this tube. And if you think about it, this is where this sodium-potassium pump is located. And it continually pumps out the sodium and pumps in the potassium. Right? So what you end up with is a higher concentration of sodium and plus charges outside, and comparatively a low charge inside, a negative charge. Right? So you basically, what you're building up is a difference in concentration of ions. And a very important concept that we talked about last time is how this transport of ions creates a energy potential, right? So if you look at this experiment with a semi-permeable membrane, then you will see that if you have more concentration of any substance that is dissolved in water, then you will have a tendency for the water to want to basically make the concentration lower, right? So basically the water would go through this membrane and go to the right side, because, well, the ions on the right side, they cannot go through. They're too big, for instance, in this example. So what you will end up with is a difference in the meniscus in this tube, which is representative of the energy that this concentration difference creates, right? So you might know, last time I also showed you the picture of water transporting up a hill, right? This is basically the same, right? So you have a higher meniscus here, which means this is potential energy, which if you would then get rid of this permeable membrane, then you would basically balance it out and get rid of this energy potential. Okay. So basically this is what we have. So the inside, or the plasma membrane of the axon, is a semi-permeable membrane, right? So usually sodium or potassium cannot go through it so well. So they need specific pores that either make them go through or that let them go through. Right? And if we look at it in the context of how these axons work, right? So if you have a plus and a minus, you have a difference in charge, which is basically like in a battery, right? So a battery functions because you have a difference in, well, in this case, it's how many electrons there are, right? So the minus side has more electrons than the plus side. So if you connect them with a wire, the electrons will flow through it and go to the plus to balance it out. And thereby we can use this flow of electrons as electricity as we know it. Right? In this case, it's not electrons. In this case, it's a difference of charge, basically, which is created not by electrons, but by positive ions, right? Sodium and potassium. And you see that with this pump that we talked about last time, the sodium-potassium pump, you create a difference in electrical potential at about -50, right? This is called the resting potential. You can maybe think of it a little bit like before, like this, right? So the pump is always creating this potential energy difference. So once a signal comes, this potential difference is released, right? And it goes to zero, even goes beyond zero, and then has to be restored again. And this propagates throughout the axon, right? From the central part to the next cell. So, and this is basically so usually the plus is outside and the minus is inside. And when this signal that we always think of as an electrical impulse is basically a depolarization. So the outside becomes negative for a short time and the inside becomes positive. Right? And after that, everything is going back to normal again. But we'll talk about that more during this workshop today, a little bit more in detail. And I hope in the end, you can fully understand how neurons work and how incredible this whole setup is to be able to transport information. Okay. So if you think about this, well, if we go back to this depolarization, you're looking at it, you can look at it like this, right? Like a curve. If you were to attach two electrodes to a membrane at 2 points, you would see the change in membrane potential when the signal comes like this, right? So first it is at rest, then a stimulus comes, right, from the signal, and then this is amplified, right? Then the voltage rises until a peak, and then the voltage falls again. It actually dips a little bit below the resting point, and then returns to the resting stage again. Right? And well, this is connected to how these transporters work, right? Because each part of this diagram, which I just showed you, corresponds to a different stage or a different basically opening and closing of the transporters which are present in the plasma membrane, right? So first, for the resting potential, I talked about, well, you always have this transport of sodium or potassium, and the other ones are not active, right? They are closed, these transporters, the passive ones. So now once this signal comes, and once it reaches a certain threshold, the sodium ones open. Right? This is called the depolarization. So that opens and all the sodium from the outside can stream in. Right? Basically, you take away the semi-permeable membrane from the U-shaped tube. And now all the water rushes back into the left side. Or in this case, the sodium rushes back into the inside of the axon. Right? This would respond to basically reaching that 0 point in the membrane potential, which is basically there is no more plus or minus on either side of the membrane. So, but since there's still a difference in concentration of other ions, it even goes over the 0 point, and more sodium than necessary would stream into the axon. And once this is done, basically what happens is the repolarization. So to balance this out, basically the sodium channel opens, and now the sodium streams to the outside, right? Because we had more of it inside before. And the repolarization happens, right? So, and after this is finished, basically the pump, both are closed, and then the pump is working again to restore this resting potential. Okay. So let's see. So this is all due to the work of these passive transporters that I want to show you today. Right? So if you think about how these passive transporters can be activated, there are actually 4 different mechanisms which are known. Which one would be, for instance, a ligate-gated channel, which would use, for instance, another molecule. If this is present and it attaches to the transporter, then the transporter opens and could go through. This actually happens in not in the axon itself, but at the contact with another neuron. Maybe, I'm not sure if I'm going to tell you about that. Maybe Pavel can tell you in the future about neurotransmitters and how these signals are modulated. It's actually a very interesting, very big topic, but I don't think I have enough time to go into all the detail in this workshop. Another one, which is very interesting, you can also have this opening be mechanically gated, right? So mechanically means with force, right? For instance, pressure. This is also very, very interesting because this is actually how we sense pressure, right? So how we feel touch in our fingers and on the skin. It's because of these mechanically gated passive transporters, right? So if there's a stretch in the membrane, both parts of the transporter are pulled open, and then certain molecules can go through. Right, so this is exactly what happens when we, for instance, would push on our finger. The cells in our fingertip, they contain these kind of mechanically gated channels, and the cells are being stretched when we basically push on our fingertip, and thereby they would allow the influx of these kind of molecules, and thereby creating a potential and a signal for us that signals us or tells us, oh, there's pressure. Very, very interesting. There are also some of them, of course, that are always open, right? That are not regulated by any second molecule or any other force. And today, the ones that we want to focus about are the voltage-gated ones, right? Voltage-gated meaning they open and close according to electrical potential, which is a very, very interesting mechanism, which is very unique compared to all other proteins. Okay, so let's look at some proteins then. Right, so we are going to look at 2 different proteins today. So some of them, like, so what is known about how these work is kind of a collective effort of analyzing all kinds of different voltage gate channels, right? Not only the one that's important for nerve cells, but there are also many other ones, right? So, for instance, not only the sodium ones, but also potassium ones, there are calcium ones, there are chloride ones, and there are even proton ones. And all that is known so far is basically, yeah, a combined effort and a combination of looking at all different models from all different kinds of animals, all different types of transporters. And in the bigger picture, it can tell us a lot about how these transporters work, right? So the first one is actually not from human, not from a nerve cell, and is also not voltage gated, right? So, but anyways, let's jump right in. So we will look at protein, right? So if you have PyMol open, actually, please tell me today who is working with PyMol, so I can kind of know how fast or how slow I would have to talk about it. Actually, I'm going to give you some time. Okay, Pavel's working with it. Okay, you're very proficient, so I don't have to be so slow. In the past, I know Ashta was working with it. Very good, very good. Also today, awesome. So, actually, let me show you the excision number again. So today we're going to type in fetch and then 5J9P. Right. Okay. Very good. 5J9P. Also, if you want to see it, it is always written on the top right of, or in the right bar of PyMol, the excision number, if you don't give it a different name. Okay, so now what are we looking at? This looks a little bit strange, right? Because if you look at it, well, it's a little bit hard to imagine, right? What it would be. And part of this is because this is only one subunit. In, I think, the first or the second workshop, I told you about that some proteins, well, they have different subunits, right? So they have different peptides that come together and form the whole protein, or the working complex. And this is one of those cases, right? This by itself cannot work. So what we have to find is basically, or we have to insert also the partners. Well, kind of symmetrical projections of this subunit, because the transporter is comprised of 4 identical subunits that make up this pore complex. So in PyMol, that's actually very, very easy. And the way to do it is, if you go to the, actually, let's first color it. I think that makes it a little bit easier. Make it C, and then you go to spectrum and you select rainbow. It looks pretty. And it also makes it easier to understand. And then you click on A in the right column. And then you click on generate symmetry mates. Right, so it will basically make a symmetrical copy of it in a 4, or it depends on what kind of distance you choose. Let's just go with the lowest one. It will create those symmetry mates within a 4 angstrom distance. Right? Okay, well. So now we have a lot of copies of the same subunit, right? And you can actually see, well, some of them, right? Some of them, they form something that looks like a structure, right? Something that would have a function. Whereas other parts here on the left, for instance, that don't look like they're very much associated with the actual protein that we want to look at. So all we have to do is just hide them. Right? So on the right, you see we made 5 copies of it. And we only need 3 of them, right? Because of 1, 2, 3, 4. We had 1 is the original and then 3 extra copies. So we have to kind of trial and error and try to figure out which are the ones that are the ones that we need or not. And this ones we did not need. So you just go to H, click on H for hide, and then click on everything. Hide. Okay, this one we needed. If you clicked on hide on accident, you can just reverse it by clicking on S for show, and then on cartoon to get it back. So what about the third one? Yep, we need that one. So we have to show it again as cartoon. How about the second one? Yep, we need that one. Oops, this show as cartoon. Okay, so this one is the one that we did not need. All right. Oops. They click on ribbon. Oh. Nope. Okay. Still have to figure out which one's which. Okay, this one. Hide ribbon. Okay. So now you have the part that is basically, well, to tell you the truth, this is also not the complete protein. There's more to it, but this is basically the part that is most interesting for us. Right? So actually, let's see. So from this, it's a little bit difficult to talk about the function. So I'm going to tell you today instead of having you guess, because having you guess is simply not fair, because even if I would look at it and I wouldn't be able to tell you exactly what is going on here. But I mean, it's easy to see if you look from the top, right? You actually, in the middle, you can see these purple spheres. Those are potassium, right? And you can see basically all the potassium is led through this red part into basically what would be the inside, or no, in this case it would be the outside of the axon. Right? So the interesting part here is basically this. This is called the pore domain. This is where the ions would go through the membrane, and which is comprised of different alpha helices. Right? We talked about it before. Very commonly you find in transmembrane proteins that they are comprised of, for instance, domains that are made up of alpha helices. Right? Very, very common. In this case, well, you have something that looks kind of pretty symmetrical, which is kind of shaped to just let potassium through. And you also can see other things. For instance, if you would think about how this would be regulated, you could imagine, for instance, that it would just close this part, right? Those 4 alpha helices. If they would change their conformation and their position towards each other, you can either, for instance, like the lens and the focus of a camera, you can either open and close it, this channel, right? So basically, it definitely has to do, the control of this channel definitely has to do with these alpha helices. Something else that you might be able to see is, well, actually, I would say this is easier to see if you only have the original. You have a part of these alpha helices that makes contact with the potassium before it goes through the pore, okay? Which would be this loop here. Which is also very interesting because these gated channels also have they are selective for what ion can go through the membrane. And this selectivity is usually due to this loop, right? The one that makes contact before going through. So I wanted to show you the structure for one, because this is basically a pretty old one. Well, I would say a relatively old one, because the function of these voltage-gated channels has been known since, I think, the late 80s. And this is basically one of the models in 1996, 1998, somewhere around that time. It was where, after the theory was made, it was actually structurally confirmed how these voltage-gated channels work, right? So this was actually very, very difficult. Because something I haven't told you about yet. Making protein structures is not very easy, right? Because, well, in this time, in the 90s, most of what was used for making these kind of structures was X-ray crystallography. And even if you don't know what it is, well, crystallography has to do with crystals, right? So, which means, well, what first had to do to get the structure is to make this protein into a crystal. I mean, okay, like for you that probably doesn't mean much, and you probably don't know. You're wondering, well, how does a protein become a crystal? But a crystal is basically just a symmetrical arrangement of multiple units. And we might look at that in the future. But what I want to just get to is that not all proteins are easily put into this crystal form and this crystal structure. Right? So one point is the bigger they get, the harder it is. And also, especially for membrane proteins, right? Transmembrane proteins, they're very, very difficult to actually put into this crystal shape, right? That's why for this time, this is all you could do, right? Structure-wise, to analyze the function of these channels. And already from this, it's super interesting what you can find out, right? And there's a lot of also like hypotheses that were made and new findings that were then later confirmed with other structures, right? With new technologies. And we're going to look at in a new model right this second. Okay, so what I want you to do is actually make a new PyMol window. See if you can actually see it. No, you cannot. Okay, so close this one, right? So we are basically done with that structure. And we're going to get the second one. from the presentation. Okay. So in this case, we are going to get the structure for so fetch, and we're going to say 6AGF. Okay. Very good. Right, and before we look at that, actually, let me tell you a little bit about how this was made. Right. So this is from 2018, I think. So very much more recently. And this one is made by CryoEM pictures, right? So this is a newer and more popular technique now, which is also needs a lot more, is more technically difficult and needs a lot of computer support. That's why in the 90s probably it wasn't really used like that. I mean, it was just in development, I think, during that time. So basically what you do is you take pictures of many different proteins of this, well, this is basically a suspension of this protein. And what you get is basically pictures like this. It doesn't look like much to you, but with the computer, I mean, you can kind of see that this is maybe one protein, right? This is another protein. This is another protein, and so on. So each dot is a different protein. And if you think about it, if you think about a fluid, they would be in different basic directions, right? They won't all look the same because they're floating and they're facing all different ways. So a computer can then analyze them and basically make like an overlay of many different of these proteins which seem to be in the same conformation, right? Or facing the same direction. And if you have enough of these pictures and enough of these overlays, you can kind of make a more and more defined model, right? I hope in the future I can tell you a little bit more in detail how this works. For now, I just want you to think about the difference in strategies and techniques that are used now compared to maybe 20 years ago, and how much more insight it can give us. Right, so what we end up with is what you see here in C, different orientations of this protein, which can then also be overlaid individually to create a 3D structure, which is phenomenal, if you ask me. Okay, and this is what we're dealing with if we're looking at the one today, right? So this is a technique which enabled not only to make pictures without making crystals, right? Which make, like for instance, making structures of transmembrane proteins a lot easier. It also made it easier to take pictures of more complicated and bigger proteins, which is a very important thing. An important fact to find out more about, well, the whole variety of proteins which are found in our cells and different organisms. So, all right, so before we can tell anything about this structure, we need to give it again color. And you see there's a lot of, well, some other molecules in there which are a little bit, well, yeah, they make it difficult to see, so we're just going to delete them. Those kinds of free molecules, they're usually found at the end of the sequence. So like we did in the past couple of times as well, try to click on display and display the sequence, right? Then you get this amino acid sequence at the top, which you can slide back and forth. And at the end, you have these extra molecules, which you can simply highlight and then right click and remove. All right. So very good. All right, what I forgot to mention, actually the last one that we looked at was not a sodium voltage gated channel. That was actually a potassium gated channel, which is not gated by voltage, but by pH. All right, so this is what they use to kind of learn about the whole mechanism of gated channels. But here, what we're looking at right now is I would say, the real deal. This is the sodium. Oh, what did I do? Okay, I just erased it. Okay, I guess I have to do it again. I clicked something and oh, I see. Okay, never mind. No, where did it go? Okay, and just erase my protein. For some reason. Let me just start new. Time fetch 6 AGF. Give it a color. Display the sequence. And erase extra molecules. Okay, so now we're back. Okay, same as before. Okay, so this is the real deal. This is a sodium voltage gated channel. All right. So structure-wise, again, you can see, well, yeah, you have a lot of alpha helices. And before we look at that in the model, let's go back to the presentation. Because these alpha helices are a very, very common motif in different voltage-gated channels. And they have different identifiers, which I want to tell you about. Right? So they're called S1 to S6. And they have different functions. So S1 to S6, those that are special are, for instance, the S4. We'll talk about that in a little bit. And you will see that the pore where actually the ion can go through is basically the linker between the S5 and the S6. You also have a N-terminal chain, and the other ones are for a little bit like structural integrity. So let's see if we can find those in the model in PyMol. Right. So what I just showed you are basically is one subunit, right? In this case, basically, if you look from the top again, you can see, well, you have 1, 2, 3, 4 similar subunits. And similar to the previous one that we looked at, you have the pore in the middle, where these kind of helices, they form like a, I don't know, like a pretty structure which like overlaps. So looking at it, you have basically 1, 2, 2, 3, 4, 5. Okay. I guess it's a little bit hard to see in this one. But I guess the identification is not that important. What's important is to see that there are some, like for instance, this is like one subunit, right? The yellow to green part. You have some alpha helices that are part of this pore, right? And you have some helices which form like an outside domain, right? So these are usually the inside is identified as the pore domain, and the other ones that are surrounding this pore domain, like this one's the green, the light blue, dark blue, and orange, they're called the voltage sensing domains. Right? How that works we will see in a second. Going back to the presentation, right? Something to note in the presentation is that for especially the alpha helix number 4 is important because it has a lot of positive amino acids, right? And the 5 is important because it links the whole thing to the pore, and then it can open and close through that. Right, so looking at it, well, this is a surface view, which we can also look at if you would like to. So instead, now we're looking at as a cartoon. But what we can also do is we can show and then show surface. So this basically shows you the surface of the protein that would be accessible from by an atom, right? I'm actually not sure if in this case it's a hydrogen or, well, you can also vary the size of the accessibility, so of the surface, but in this case this is like the standard what we're using right now. So what you can see is, well, if you look at this protein, there's one hole in the middle, which is the pore. Oh, sorry, you don't see it. Right? So, because you didn't see it, I'm going to show it again. So in this case, you can click on show, and then you click on surface, and then it will create this colorful picture. Right? So you can see in the middle is the pore. Right? If you want, actually, you can pretend to be a sodium and fly through it in a way, which is, I find it very interesting because you can go really deep and inside the protein and basically have like a first-person view of what it would be to be a sodium that wants to go through it. And something that you can see, for instance, there are different the space is a little bit different inside, right? So there are some areas that are more accessible. It's easier to go through, and there are some that are limiting, right? This limiting area, for instance, would be, this is the top view, right? From the top, would be a little bit if you go to the inside of this pore, where it has like I would say the smallest diameter. This is where it basically selects which ion to let through. If it's a sodium or a potassium. This is the identifying area. Right? And after that is done, it can basically pass through and get out on the other side. Something that that I want to note here, which you can do in PyMol, but I haven't gotten to work yet, You can give this also a surface which is colored by how basically polar it is, right? So if it's red, like in the right representation, it would mean that it's more electronegative, which means that it would attract positive ions. And it's important to mention here that the whole pore is very electronegative. So it kind of attracts the sodium ions to go through it by having a overall negative charge. Right? Okay, very good. So let's see. One more time in the model. Looking over it, you have this pore, you have the identifying area, you have the side where it comes out, and yeah, that's it. Let's look at a different part. So what we're going to do is we're going to hide the surface. Right, so H, hide surface, and then you're back to the cartoon representation. Right, and you kind of can see a similar thing, right? But of course, with surface, it's a little bit more easy to imagine where the actual access point for the ion is. So now we're going to take a look a little bit more, not on the pore domain, but on the voltage sensing domain, because this is where the whole control mechanism sits, right? For this, let me look. There's actually a amino acid sequence which is very, very important, which is the same for all 4 subunits, right? So in this case, the subunits are labeled as VSD, which stands for voltage sensing domain. And since we have 4 symmetrical units, they're units 1, 2, 3, and 4. And this is their respective amino acid sequence. They're a little bit different in some parts, but the important part are those R's, basically, right? And you can also see them here in a different representation than we have. They're basically stacked on top of each other on an alpha helix. In this case, the S4, right? Which I showed you in the model at the beginning. So let's try to find this in our model, right? In our PyMol. And for this, all you have to do is find basically the amino acid with the respective number, right? So R1129, for instance, one of them. Let me show you what R1129. So if you go back to PyMol, so you have the whole sequence at the top. So R1129. Here it is. Here's another R, here's another R, and here's another R. Apparently, in this case, also the K is important. So we're going to highlight that as well. Let's see, is there another R? Yep, that's also this one. Maybe that as well. So this is a selection that we just made. So we just clicked on them in the sequence, and we can give them a separate name to be easier selectable later. So I'm just going to rename it to R's. And I'm going to show them as licorice, right? And I'm also going to give them a different color so they're visible more easily. I'm going to make them red. Okay. Okay, and we're also going to center on them, so it makes it easier to navigate. So this is alpha helix number 4, the most important one in this protein, because this is the part where protein senses the voltage. It's connected to the pore domain, right? Through this yellow and green helix. And those R's, they're all positively charged amino acids. Which means if you remember what we talked about, the resting potential of the axon, we talked about that the outside is positive and the inside is negative, right? So in this case, well, if these amino acids are positively charged and the outside is positive, well, then this alpha helix would be basically pushed inside or into this position, right? So this is the off state. I mean, it looks kind of open, but this is basically supposed to be the closed state of this protein. Now, we talked about when a signal comes, right? A signal, in this case, means a depolarization of the membrane. Depolarization meaning plus switches to minus. So what that means, the force of the positive charge on the outside is reduced, right? So if it is reduced, what happens? Well, this alpha helix can then pop out, basically. It can pop out and change its conformation, right? Just moving upwards by like basically one step. And like a plug, basically it carries with it this part or this alpha helix and pulls on it, thereby opening this whole pore complex, right? So this pulls up, and thereby this one gets stretched and pulled also in direction outward, and thereby opening this pore. This is the whole process, right? Once it gets repolarized, right, so the plus charge gets stronger again on the outside, and basically it gets pushed back in and the pore closes again. Okay, thereby stopping the flow of the sodium ions, right? If it pops out, it's opened and sodium ions flow in. So let's look at it in the presentation. So in this case, well, here are the amino acid sequences again, right? So because of the 4 subunits, they all have the same alpha helix with the same sequence that reacts to the voltage. And here you can see them basically in a different representation. It's a little bit more complicated, I think, to understand. And here is basically a little bit focus on the linker, how this change in conformation is then translated into opening of the pore. Let's see. So if you look at it from a more schematic representation, right? What we have highlighted on the left side is, in yellow is the voltage sensing domain, right? Which was the part on the outside. We have the positively charged S4 alpha helix and the inside pore domain here in green, right? And represented here on the right, again, similar to what we saw. So now, if the voltage comes, it can be activated one by one. but it's only fully open if all 4 of these S4 helixes pop out, basically, out of the structure. Oh, that's 1, 2. Right? Okay. So I hope that kind of shows you how this whole thing works. Another thing which is important to mention, if I go back to one of the previous representations, something that's very interesting, especially about sodium gated channels, sodium voltage gated channels, is they have a well, it's called a ball and chain, right? In their structure. We couldn't see that on our PyMol, unfortunately, because that's also something that has not been characterized yet and been like analyzed yet in either crystallography or with cryo-EM, because this is a relatively unstructured area and is hard to see. So, but the function of this is represented here. It basically acts like a plug to the whole pore, right? A plug that stops the flow of sodium before the actual channel is closed, right? So in this representation, you see the pore domain in green, and you see the voltage sensing domain in blue with the specific alpha helix. And what you can see is also here, in this case schematically, this ball and chain N-terminal domain. So what happens when it gets depolarized, it opens, the sodium flows in, this ball part is actually attracted to the opening of the pore. So once it opens, it actually, there's a chance it will actually attach to it, get in contact, and then close it. So no sodium can flow through anymore. Now you're asking yourself, so what's the point of that, right? So why is there something that would close the channel when it's supposed to be open because of the voltage difference? This is actually a super smart design, and you have to look at it in the context of the whole signal going through the axon, right? So what you have to remember is kind of there are 3 different states this whole complex can be in. You have the closed state, right? At the beginning, you have the open state once the signal comes, and you have the inactivated state, when basically the chain and ball block the pore. So if you think of it, if you're looking at the axon potential, right? So first, you have the pumping that we talked about, right? That generates the resting potential. You have the threshold. So now you actually can understand what the threshold is, because the threshold is the voltage, like the voltage change that is necessary for this alpha helix to pop out and thereby opening the channel, right? So at this point, basically the sodium channel opens and says, okay, all the ions come in, right? So in this case, the membrane potential rises and it actually gets depolarized. But then, well, what happens is the potassium channel gets open, so the voltage goes in the other direction. What would happen if you weren't to close this sodium channel fast enough? What would happen is probably the potential would not return as fast as it is in this case, right? Because still there would be potassium flowing in, potassium flowing out and sodium flowing in, meaning, well, basically it would stay at 0. right? So instead of repolarizing because of the flow of the potassium, it would actually, if the sodium channel would stay open, it would actually, well, stay at 0, right? Because to kind of, the sodium wants to always neutralize this negative difference. So basically this ball and chain closes it so a repolarization can happen. Okay. And okay, I know it was a little bit complicated and we went into details a lot for today. So at the end, I want to put this all into context, right? All the explanations that I did right now, I actually put into a very, very well-made model, and this is the link that I shared today from the, I think it's the Ministry of Education or whatever it's called in America from Colorado. They always make these kind of online, freely accessible models of different physical and biological and other phenomena. In this case, they also made one about the axon potential. And I would like you to follow this link, right? I'm also going to follow it. And this is what you would look at. So this is a simulation of all the things that we talked about last time and this time, right? So you have different parts. What you're looking at is a cross-section of an axon. So this cross-section is made up of, what I said, sodium-gated channels and potassium-gated channels. What they didn't show here is the pump we talked about last time, because this is like right now, if you look at what's the potential charge, which you can opening by clicking on the check mark. So right now, this is basically the resting potential, what we're looking at. So if you can actually zoom in, you can zoom out. What you can see is you have sodium, which are the orange dots, and you have potassium, which is the green diamonds, basically. You have more potassium on the inside, and you have more sodium on the outside. Okay, very good. So there's already something happening if you look at it. If you zoom in, you can already see, well, there are some leak channels, right? So there's always some sodium going in and some potassium going out, and there's always a little bit of leakage between the membranes. Actually, I'm not a neuroscientist, so I'm not 100% sure. I forgot to look it up why that is. I'm sure that Pavel can tell you why. I would, if I were to guess, it is because to keep a certain, probably also physiological ion concentration, but also to keep the membrane potential at a certain point. So there's always like a little bit leakage. So the pump is always working. It's keeping this resting potential at the comfortable minus 75 millivolt. And there's always a little bit liquid. Yeah, okay, to avoid hyperpolarization. Yeah. Okay, very good. So there's always a little bit leakage of these ions through the membrane to keep it from going too low in the voltage difference. And then you have basically the sodium-gated channels, potassium-gated channels, green, red, in a closed state. So what you can do in this simulation is basically You can, for instance, also look at the charges, right? So you have positive on the outside, negative on the inside. You can look at concentrations, which are, I think, not as interesting. But what you can do is you can stimulate the neuron. Right? Stimulation means a potential is built up and then travels through the axon. So if you click on that, you'll see like a purple ring comes. And this is basically what happens inside the axon, right? So this was very fast. So let's reset everything. You can zoom in again. You can show the potential charge, chart the charges, and let's do a little bit more in slow motion. Because, well, you know, nerves work really, really fast, right? So in reality, it's even faster than shown here, how these channels work. I think it all happens within 0.5 milliseconds. This opening and closing cycle. Okay, so let's stimulate one more time, and let's look at what the channels are doing. Right? So stimulate the neuron, then the potential comes. The sodium channels open. They close themselves up with the ball and chain. The sodium goes out. Sodium channel closes. Potassium goes out. The potassium channel closes. And the sodium channel closes as well. Okay. Still very fast. I clicked on the slow motion on the bottom, but still everything is happening very fast. But that's the cool thing about this model. You can always do it again. Alright, so we set another signal. Opening, depolarization, minus outside, plus inside. Closing, like inactivated state. Potassium goes out, repolarization, closing, and we start again from the resting potential. Okay. One more time. Opening, closing, and so on. Okay, very good. So I hope you can play with that for a little bit more. You can make it faster. You can look at it from different angles. And this is this is probably more closer to the speed that it actually happens. Signal comes, open, close, and depolarization, repolarization, and finish. All right, so very, very quick. Yes. All right. With that, I hope you can play with the simulation a little bit more to make it more clear to you what is happening. Otherwise, I think the workshop, well, all I wanted to tell you today is done. If you have any questions, please ask in the YouTube chat. I'm going to give you a little bit of time because there's a little bit of delay, and then I hope I can answer some of your questions. If not, of course, you can always ask them to me personally on Slack or on Discord. Let's see what kind of questions we have. I'm also going to play with it while I'm waiting. How is the neuron stimulated? Okay. Well, this may be like a very big side topic if you were to look at it. Like some of it can be chemical, right? So some of for instance, what we looked at, some of the potential can be generated by actually opening channels in response to certain neurotransmitters, right? So if they bind to these channels, they can basically force them open and thereby creating this whole depolarization repolarization chain, and then this just goes on through the axon. At least that's the one I know of. Please, Pavel, enlighten me to what other way there is to stimulate a neuron. I don't know in what way it could happen internally in the cell. I thought it would always be a somewhat external stimulus that is necessary. Good enough. Okay. Right. Yes. So yeah, we talked about in the beginning at the different ways that gated channels can be opened. So if they're opened not by voltage but by, for instance, other chemicals or molecules or neurotransmitters, then a potential can be generated. It's actually very interesting. I would like to know more about that. What reasons can be that neurons start to fire? If it's not something externally, maybe could they do it maybe with internal signal? Any other questions from students maybe? Okay. Very good. So I hope I was able to convey this amazing machinery of how nerves work and how these kind of signals that we always think about as like electrical signals that like pass through our brain and make us who we are all based on the work of these molecular machines, which are in the plasma membranes of axons. They open and close, and that's the whole magic behind it. Right? It's not something supernatural. It's something, if we just research it, has a very, very intricate logical explanation, which I think really amazing. Okay. And with that, I hope I will see you guys next week again. And actually, I'm not sure. I think we're going to talk about neurons a little bit more. Not about a specific protein per se. I think it might be time that we actually look at the ways that these kind of protein structures and 3D models are created. Because I think it's a very interesting part of, and also an important part of this workshop, to understand what you have been looking at the past workshops. With that, thanks for coming, and I will see you guys next time. Bye bye.